All right, welcome back to the deep dive. Today, we're going to be taking a deep dive into something pretty fundamental to life as we know it. We're going to be talking about photosystem,
ASI.
or PSI for short.
Yes.
Most people will probably remember from their biology classes that photosynthesis involves two systems, and PSI, which is what we're talking about today, is found in algae and plants and cyanobacteria, and basically it uses light to move electrons around,
Yeah.
ultimately creating NADPH. NADPH is a really important energy carrier. It fuels ATP production. And what's really interesting to me right off the bat is that even though PSI functions after photosystem II in the process,
Right.
it was actually discovered first.
Yeah.
Kind of a weird quirk of scientific history there.
Yeah. It speaks to how we often, we learn things in pieces and then we put them together later.
Yeah, exactly.
Sometimes we find the end of the story before we find the beginning.
Right, right. And this is one of those cases where it's kind of like
Absolutely.
we discovered how this thing worked later on. So what's really interesting about PSI is it's quite complex. Just thinking about how all this stuff works together, it's really amazing.
Yeah. What's striking about PSI is just from the get-go how many components are involved. There's over 110 cofactors
Wow.
involved in this system. And that really just speaks to the fine-tuning, the level of controlled machinery that needs to be in place in order for this to work properly.
So that's our mission today, is to understand how all those 110 pieces and parts work together.
Yeah.
And what their functions are.
Absolutely.
And maybe we can even touch on its ancient origins. I mean, this is something that's obviously very fundamental to life as we know it.
Yeah.
So let's start with some history, just to get us warmed up.
Yeah, great idea.
Yeah.
So the initial observations, people were kind of noticing things going on in the 1950s, but they weren't quite sure how it was working. But the idea of photosystem I and T, the formal kind of concept, that came together in 1960, thanks to Louis Dyson. Going to put that together. And then in that same year, Bendel and Hill proposed this concept of these systems working in series, one after the other, which was confirmed experimentally just a year later in 1961.
Wow.
So that, shows you how quickly sometimes things can progress in science.
Absolutely.
Where you have the proposal, and then boom, confirmation a year later.
And that was in the 60s, right?
Yeah.
1964.
So, science was cooking back then.
Yeah.
Okay, so let's get into the nuts and bolts now of PSI. We know that at its core, there are these two main protein subunits.
Yes.
Called PSA and SIB.
Right.
Now, these guys are embedded in the membranes of these things called thylakoids, which is where all the action happens, right?
Yeah, that's right. They're integral membrane proteins. So they're actually embedded within the thylakoid membrane, which is inside chloroplasts.
Right.
And they're, as you said, they're really essential because they bind the key electron transfer cofactors. So these are like the main framework on which everything else is built.
Yeah, that's a good way to think about it. They're the scaffolding that holds it all together.
Got it. Okay. So now let's talk about the other components.
Right. So kind of extending that chain, we have phanoliphobe.
Okay.
And those are also iron-sulfur clusters. And specifically, they are four sulfur clusters, which is just referring to the particular arrangement of the iron and sulfur atoms.
Okay.
It allows them to easily gain and lose electrons, which is essential for the process. And these are located on a protein called SAC.
Okay.
Which is associated with that SAC-SAB core.
Got it. So these iron-sulfur clusters, they're kind of like
They're like little relay stations.
relay stations, yeah.
Yeah, passing the electrons along.
So PSI needs to grab light energy in order to kick off this whole process.
Right.
Right, that's where the antenna complex comes in.
Exactly.
So can you tell us a little bit about that?
Yeah. So it's this collection of chlorophyll and carotenoid molecules. You can almost think of them as like tiny little antennas.
Little solar panels?
Like little solar panels, yeah, attached to proteins.
Okay.
And their job is to capture those photons of light, those little packets of energy, and then funnel that energy to the reaction center. And what's interesting is that the number of these kind of panels varies across organisms. So, for example, in cyanobacteria, you might have 90 chlorophyll molecules in the antenna complex.
Okay.
But in spinach chloroplasts, you might have more like 120. So it's not like a fixed number.
It's not a one-size-fits-all kind of deal?
No. Different organisms have adapted to different light conditions.
Right.
And that's reflected in the size of their antenna complex.
Got it. So all that energy is being funneled to this thing called the P700 reaction center.
Exactly.
Which is like the heart of the operation here.
Yes.
Tell us about that.
Yeah, so this is a special type of chlorophyll, A, and it absorbs light best around 700 nanometers.
Mm.
So when it absorbs enough energy, it gets excited,
Yeah.
and we call it P700 star.
Okay.
Its electrons jump to a higher energy level. They're ready to rock and roll, ready to start that electron transport chain.
Right.
And so this excited state, the P700 star, that's what initiates the whole movement of electrons.
Okay.
And it's really important because that's what allows the whole system to capture energy from light and use it to power those downstream reactions.
So once that electron gets excited and starts moving down the chain, what happens?
Well, it moves to those early acceptors, A0 and A1, which are modified chlorophylls.
Okay.
Then it goes to phylloquinone, which is also known as vitamin K1.
Okay.
Then it goes through this series of iron-sulfur complexes,
Okay.
Fs, Fe, and Fab.
And those are those relay stations we talked about earlier?
Exactly, those little relay stations,
Yeah.
passing those high-energy electrons along.
Okay.
And there's some debate, actually, about the exact order of electron transfer between those iron-sulfur clusters.
Okay.
But the overall process is still, pretty well understood.
Got it. So what happens after those iron-sulfur clusters?
So eventually, those electrons end up on ferredoxin, which is a protein that then carries those electrons to the enzyme ferredoxin NADP plus reductase.
Oh, wow. That's a mouthful.
Yeah. It's got a long name, but its job is basically to convert NADP plus to NADPH.
Right. And NADPH is one of the important energy carriers that we talked about earlier.
Exactly.
Okay. So we've got this whole chain of electron transfer happening, but we also need to replenish those electrons that are getting lost from the P700 reaction center.
Right. That's where plastocyanin comes in.
Okay.
It acts like a little delivery truck, bringing electrons to the oxidized P700 plus.
So it's like the recycling truck that comes along and picks up the empty bottles.
Exactly. Keeps the whole cycle going.
Okay. So is that all the players, or
Okay.
are there any other important components?
Well, there's also this protein domain called YCA4,
Okay.
and that's actually crucial for assembling the entire PSI complex.
Mhm.
So it's not directly involved in electron transfer, but it's really important for putting the whole thing together.
Got it. So it's like the construction crew that comes in and builds the factory.
Yeah, that's a good analogy.
So we've talked about all these different components of PSI, but now I'm curious about its evolutionary history.
Yeah.
Where did this complex system come from?
Well, there's some pretty compelling evidence that links PSI to the photosystems of green sulfur bacteria.
That's interesting. How so?
Well, first of all, both PSI and green sulfur bacteria have a low enough redox potential to reduce ferredoxin,
Okay.
which is important for their function. And they both use iron-sulfur proteins in their electron-accepting centers.
So there are some structural similarities there.
Right, there are some structural and functional similarities. But what's even more striking is that green sulfur bacteria actually have all the same electron transport chain cofactors as PSI.
Oh, wow.
So that suggests a really deep evolutionary connection. It suggests that they might have shared a common ancestor.
So PSI might have evolved from something similar to the photosynthetic machinery in these bacteria.
Yeah, that's the idea.
That's amazing. So we've learned a lot about PSI today. We've talked about its history, its components, how it functions, and even its evolutionary origins. It's just incredible to think about all those different pieces working together to capture light energy and convert it into chemical energy that can be used by the cell.
Yeah, absolutely. And as we said at the beginning, the sheer number of components in PSI is mind-boggling.
Over 100, right?
Over 100, yeah.
Yeah.
Proteins, lipids, pigments, coenzymes, and cofactors. It's a symphony of molecular machinery.
And all of that working together to achieve this one really important task.
Exactly.
So I think we've covered a lot of ground today, but I want to leave our listeners with a final thought to ponder. Considering these deep similarities between PSI and the simpler photosynthetic machinery in green sulfur bacteria, what fundamental insights into biological energy conversion could studying these more basic systems provide? And could that knowledge lead us to entirely new bio-inspired technologies?
That's a great question, and one that researchers are actively exploring.
So it seems like the study of photosynthesis is not just about understanding plants.
No, not at all.
It's about understanding the fundamental principles of energy conversion in biological systems
Right.
and how those principles might be applied to solve some of the challenges we face today.
Absolutely.
Well, that's all the time we have for today, but I want to thank you for joining us on this deep dive into Photosystem I. It's been a fascinating journey.
My pleasure.
And we'll see you next time on The Deep Dive.
See you then.